Hello, we are the extractors.
And in this episode, we're going to extract DNA from our saliva.
In this episode, we will be using
The first step is to pour half a cup of water. Once you do that, you have to grab a bit of salt. You have to get 1/16 of a teaspoon for this experiment because you can't actually gargle one full cup of water, so we have to divide everything by 2. So you'll be doing this twice. So once you get the 1/16 of salt, you put it into the water, and then you have to gargle that for 1 complete minute. The next step is to gargle the water. You have to make sure you get all of it and gargle for 1 complete minute. Have some handkerchiefs close by in case you make a spill.
So after you have your glass of spit already spat out, what you have to do is put it in another separate cup through a strainer. So that's what I'm going to do right now.
And now we need to mix 2 teaspoons of detergent inside the mixture. And so we're going to need to pour 2 teaspoons. And we're going to be using a normal regular teaspoon to measure. You might need to use a bowl because if you spill, which you will, you're going to need a place to catch the fallen detergent.
So that's 1.
That's fine. Like that.
Come on. That's 2.
And that's 2. Now you want to mix it around a bit, not too much, just so it's there. And now,
Just for 5 to 10 minutes.
now you want to let the mixture sit for about 5 to 10 minutes so everything gets settled. And see you then.
Once you've let the solution sit for 5 to 10 minutes, you want to grab a small test tube, but in this case we'll be using a shot glass. Now because our cup is too big to actually pour into this, we're going to be using a smaller cup. You want to grab a bit of the solution and pour it in just to 1/3. Just fill up the small test tube for 1/3 of it. Doesn't matter if you get a couple spills since you will have more of it. Your final result should be this.
Up, since we have the beak shot glass filled, where you have to put the enzymes in it. Now for the enzymes, we're going to be using meat tenderizer. Meat tenderizer is pretty easy to get. It's useful for this experiment. Now, what we're going to do is grab a pinch of the enzyme, or meat tenderizer, and we're going to just put a little pinch, just a tiny pinch inside the solution, like so. Yeah, that's much. Now, you're supposed to stir it, and you're supposed to stir it, but if you stir it, you're supposed to do it really slow, because if you do it really fast, your DNA will be really hard to see. That's good.
Hello, this is stage number 4. So now what you want to do is grab a little bit of rubbing alcohol, put it in a cup so you don't make a lot of spilling, and you want to make sure to tilt the cup full of the solution a bit and pour the rubbing alcohol very lightly, about the same amount of the solution. So it has to be like half solution, half rubbing alcohol. So I'd say about that much is fine. Your end product should be like this after step 4.
All that, after about a minute, you should see that the alcohol doesn't really mix with it. As you see, it's blue down here from the original mixture and white up here from the alcohol. And if you look between that, you see that white layer? That's your DNA. That is the DNA you just extracted from your saliva.
Wow! Whoa!
As you all may know, DNA is a small, stringy molecule. The salt you added in step 1 helps it stick together. So actually what you see right here, the stringy part, is Gus's DNA. Because he spit out the water. See that? That layer of stringy stuff? That's the DNA.
Now, normally the DNA would dissolve in the water, but since we added the rubbing alcohol just keeps it from, it just untangles all of it so it stays there, and it doesn't dissolve.
Now what's interesting is that you can get this DNA out of here. What you do is you grab a stick, we're using this, a drink mixer. You just go in, get it out, and just put it on a small Petri dish. So that you can conserve your DNA. Just like that.
And then you can use it for cloning.
Yeah.
You've just been